Okay, well, we have a few loose ends to tie up between now and for next Tuesday when we'll have our test. I assume everybody got my email about that. Okay, good. So, I think lipids are where we left off, and so that's where we'll start. Remember that lipids are nonpolar, so they're going to be insoluble in water. And if they are nonpolar, what kind of interparticle forces would you find between closely spaced adjacent molecules of lipids? Dispersion forces, right, because it's nonpolar. All right, the common ones are triglycerides, phospholipids, and cholesterol. And so, without further ado, let's take the triglycerides. Tri, of course, means three of something. It's going to be three fatty acids, or what the somethings are. And they're in combination with a molecule of glycerol. So you have glycerol or glycerin, it's the same thing, and three fatty acids. Now, here's kind of the general overall organization. Glycerol just is 3-carbon alcohol, so if you had glycerol by itself, it would just have an OH over here on the end. But that is where we hang on our three fatty acids, and the fatty acids are extremely nonpolar. So they make this whole structure then nonpolar, and so therefore it's not going to be soluble in water. Now, what is a fatty acid? Well, they are a long carbon chain molecule. Lots of carbons and hydrogens. And out on the end, they have a particular functional group called a carboxyl group. And anything that has a carboxyl group on it is a carboxylic acid. And so when it comes to a carboxyl group, this is what it looks like. And carboxyl group can act as an acid because what do acids do when you put them in aqueous solution? What do they give up? Hydrogen ions or protons, right. Well, there's a hydrogen ion or proton it can give up. So, there you go. Now, we don't really have to worry so much about these because they're going to be water insoluble. But they do have that carboxyl group on the end, so they have the potential of acting as an acid, because anything that has that on an end, well, that can act as an acid. And we'll see it again with another class of compounds here in a bit. All right, so there we go. So there's the fatty acid. It's a carboxylic acid. And when it comes to classifying fatty acids, we have three kinds, and this is the thing you really need to put the star by. We can have what are called saturated fatty acids, unsaturated, and polyunsaturated. Now, saturated. They're saturated with hydrogen. In other words, they contain the maximum number of hydrogens that you can add. All you're going to find between the carbons in a saturated fatty acid are single bonds. Okay? And that's the thing really to look for, I think. If you look at it and see that you only have single bonds between the carbons, you're looking at a saturated fatty acid because it's going to contain the maximum number of hydrogens that you can possibly add onto that. And a hydrocarbon chain is nonpolar. And that's going to account for the nonpolar characteristic, really, of these triglycerides. Unsaturated. It has one double bond. So between two adjacent carbons, there's a double bond in there. Now, obviously, if you throw a double bond in, you've got to take two hydrogens out. So it has less than the maximum number of hydrogens. The important thing here is this. When you throw in the double bond, you put a bend in that hydrocarbon chain. If it's a saturated chain, they're all kind of pretty much in a row. It's a nice, straight, zigzaggy chain. But if you throw in a double bond, you put a bend in it. Now, when you go putting bends in these things, you can't pack them as close together. So when you can't pack the molecules as close together, what's going to happen to the strength of the dispersion forces between the adjacent molecules? Are they going to get stronger or are they going to get weaker? Weaker, because there's more space between them, right? Polyunsaturated, they're going to have 2 or more double bonds, all right? 2 or more between the carbons. So even fewer than the maximum number of hydrogens that you can put in there. Well, every time you throw in a double bond, you bend the chain a little bit more. And so there you go. And you weaken those dispersion forces even more. All right, now let's take a look at this saucy little tidbit, which I think now should make a little more sense. And that is that lipids that are made up of just saturated fatty acids, in other words, triglycerides that just have 3 saturated fatty acids, they're likely to be solids at room temperature. So what would be a good example of something that would show that? Well, how about lard, pork fat? Remember Emeril Lagasse, what was one of his pet expressions? Pork fat rules, remember that? You don't? Well, he's been off TV for a while. All right, beef tallow. You know, like that fat that's on a steak or a roast. I mean, you know, that sort of stuff. Pretty solid, right? Saturated in fatty acids. All right, but now what happens when we go with unsaturated and polyunsaturated? Well, remember now, if they're made up of unsaturated fatty acids or polyunsaturated fatty acids, there's going to be more space between the adjacent molecules of triglyceride. The dispersion forces are going to be weaker. These are going to be liquids at room temperature, or what we commonly call oils. So if you were to take something like canola oil, for example, and you know it's a liquid at room temperature, it's going to have a lot of unsaturated, polyunsaturated fatty acids in it. Still a triglyceride, yes. Glycerol and 3 fatty acids, but they're not saturated fatty acids. That's the difference. All right, so let's take a look at this. This is an example of a saturated fatty acid. What do you notice about the bonds between the adjacent carbons? They're all single bonds. So that would contain the maximum number of hydrogens that you can put with those carbons. Now, if we take a look at this next one, which is actually mislabeled, what have we got? We've got 2 double bonds between a couple of the adjacent carbons. And when you throw in a double bond, you've got to take out 2 hydrogens. All right, so it doesn't have as many. But notice what else happens, and this is probably the more important thing. What happens to that? You see how straight that was. What's happening to this one when we go throwing in the double bonds? It bends. Okay? There you go. Now, why is this mislabeled? It should be polyunsaturated, right, because it's got 2 double bonds in it. All right? Now, don't worry, because here's one that is unsaturated right here. There's a triglyceride, and we have over here the glycerol, and then we have the 3 fatty acids, one of which is unsaturated. Why is it unsaturated? It has a double bond between 2 adjacent carbons. Just one double bond. But again, you notice that causes a bit of a bend. So this one, if you had a bunch of these, they would have weaker dispersion forces than if you made up a triglyceride that just had 3 saturated fatty acids in it, because you couldn't pack these quite as close together. The dispersion forces are going to be a little weaker. And if this one is a solid at room temperature, and it may be, it would have a very low melting point, okay, as opposed to one that had just strictly saturated fatty acids. Well, they would have a higher melting point, okay? And you notice that, I'm sure, with fats, how some of them melt at very low temperatures, and turn up the heat very high to get them to melt, and others They just don't, typically, even when you're practically incinerating them on the grill. But anyway, there we go. All right, now where do we find triglycerides? Well, those are what are typically stored in adipose tissue. And I assume you've all seen adipose tissue in lab, right? The adipose cells or adipocytes have that big empty vacuole in them. Well, in life, what would have been there would have largely been triglyceride and maybe a bit of cholesterol, okay? And it all got dissolved away when the tissue was processed. So I bet you could have guessed in a million years what kind of solvents would have been involved there. If you're going to dissolve away the fat, would have been water? No. Something very nonpolar is exactly right. Yes. Okay. See how simple that is. All right, phospholipids. These are the major class of lipid that make up biologic membranes. So when we get to talking about cell membranes, as we will before long, we're going to see that they are largely made of phospholipids. In fact, 2 layers of phospholipids. All right, what is their main claim to fame? Well, what about their composition? What have they got? They've got glycerol again, all right? And this time only 2 fatty acids, not 3. But taking the place of one of those fatty acids is a phosphate group, all right? So now, what elements are we going to find in a phospholipid? Well, for inspiration, let's back up here. Let's go back to this one. Now, there's a triglyceride. Now, what elements do you see in it? Right, carbon, hydrogen, and oxygen. Now, what is the big difference between this and a carbohydrate? What was the thing that really gave away carbohydrates? Yeah, there are carbon, hydrogen, and oxygen as well. But what was the big thing that gave them away? And you're not going to see it here. 2-to-1 ratio between hydrogen and oxygen. Do we have a 2-to-1 ratio between hydrogen and oxygen here? No, we don't. It looks like in that thing I only see 6 oxygens and gobs of hydrogens. Right? Now, kick it up a notch. And if we go to phospholipids, what are we going to find? Well, here's kind of a graphic representation of it. There's the glycerol. The fatty acids, again, would just be carbon and hydrogen, and throw in an oxygen or so. And here's a phosphate, and then here's something that some of them, not all of them have. It's what we could call an amino, A-M-I-N-O, amino alcohol. Now, what elements do you see here? Carbon, hydrogen, oxygen, nitrogen, and phosphorus, right? At a minimum. Carbon, hydrogen, oxygen, phosphorus. Sometimes nitrogen. All right? Are the hydrogens and oxygens going to be in a 2-to-1 ratio this time? Hardly. You're going to have gobs of hydrogens, very few oxygens. All right? How many phosphorus in this thing? Just 1. Well, see, that's simple, isn't it? Okay? See, they just give themselves away. So phospholipids are going to have carbon, hydrogen, oxygen, and phosphorus. And sometimes they'll have nitrogen as well. All right? Now, here's another trick about phospholipids. This area where we find the phosphate and perhaps the amino alcohol, that's going to have a charge on it. That's going to turn out to be polar. And we're going to refer to that end of the molecule as the polar head. So the phosphate end of the molecule is polar, and we call it the polar head. It actually interacts with water. Is it water soluble? Well, no, not really. But it will align itself with water, and it can hydrogen bond with water. All right? reacted with water? Not at all. That area of the molecule we're going to call the nonpolar tail. Nonpolar tail. So if we look at this representation of a fatty acid, excuse me, of a phospholipid, here's the head, the polar head, that's the phosphate end, and the glycerol back in here too. And then what are these two things? These are the nonpolar tails. What are they? They're the remnants of the two fatty acids. And they're very nonpolar. Now, because of that, you're going to see then when we look at cell membranes how these things align themselves. Guess what's going to be on the surface of the membrane? Is it likely to be the polar head or the nonpolar tail? Because remember, on the surface of the membrane, there's going to be lots of water molecules hanging around. I think the polar head's going to be on the surface, and the tails are going to be down in the center to get away from that water. Well, chat about that when we get to that soon. Here we have a couple of models of some phospholipids. And so you'll notice in this one, there's the polar head, there's the glycerol part, and then here are those non-polar tails that are made up of the fatty acids. All right? And you notice these are fairly linear. All right? But if we take a look at this next one, look at that. I wonder what kind of a fatty acid that's likely to be. I would say it's probably polyunsaturated. It's at least unsaturated, more likely not polyunsaturated because of, look how much of a bend you've got in that. All right, here again, polar head, glycerol, non-polar tails, phospholipid. And in the polar head, we're going to have, of course, a phosphate group at a minute. Now, the sterols. These are waxy-like substances that contain 4 fused carbon rings. Now, wax. What is the physical state of wax at room temperature, I wonder? It's a solid. Yes. And it's going to be, again, a non-polar covalent substance, right? What kind of interparticle forces between closely spaced adjacent molecules, I wonder? Yeah, dispersion forces, of course. All right. Now, those 4 fused carbon rings. Let's take a look at that. And here it is. And you just have to imagine at each one of these points there's a carbon. And that is the general structure of a steroid. It does have the name. Let me write that up here for you. Whenever you see that structure, that is referred to as a sterane skeleton. Not bones, but carbons and hydrogens. All right, here we go. So those 4 fused carbon rings, that's called a sterane skeleton. And that's going to be the basis of not only cholesterol, but any sterol type lipids. And we'll just take a quick look here at cholesterol. And here again, don't go into heart failure. You're not going to be required to draw this out from memory. But if we back up, we note that the most common example of a sterol in our cells and in any animal cell is cholesterol. All right? And cholesterol's got a number of uses. All right, here is cholesterol. And here they've actually thrown in the carbons and hydrogens. And you notice other than carbon and hydrogen, there's just one little lonely oxygen all over here by its lonesome. Now, where are you going to find cholesterol? Well, one of the places you're going to find it, it's also in cell membranes. Now, the majority of what make up cell membranes are the phospholipids. But there's going to be some cholesterol in there as well. Another place you can find it, there will be some of this stored with triglyceride and adipose tissue. And another thing that we want to note about cholesterol is it is the basis for the synthesis or production of sex hormones. The sex hormones are all derivatives of cholesterol. So the body starts with cholesterol and then transforms it into those hormones. And any of the steroid-based hormones are really derived from cholesterol. So there we go. And again, none of these lovely charts you can't read. But triglycerides, what have we got? 3 fatty acids and a glycerol. What elements would there be in a triglyceride then? Yep, carbon, hydrogen, and oxygen, but what about the hydrogen-oxygen ratio this time? It's not 2 to 1, right? There are only about 6 oxygens in there, right? Maybe a few others depending on what's making it up, but in any event, there you go. Where do you find it? Stored in adipose tissue. That's the big claim to fame for triglyceride. Supposedly, and there is a lot of energy stored in triglyceride. The trick of it is once you store it away, you never break it down. It seems, right? I don't know about you, but anyway. Phospholipids, what are they made of? Glycerol, 2 fatty acids, and a phosphate, and maybe an amino alcohol. So what elements? Carbon, hydrogen. Notice how carbon and hydrogen all go together so well. Carbon, hydrogen, oxygen, phosphorus, and maybe a nitrogen. And again, you're not going to find a 2 to 1 ratio between hydrogen and oxygen. Okay? Now, where do you find phospholipids? What was their main claim to fame? Cell membranes, right. Okay? And then we have sterols. And the sterols, 4 fused carbon rings. What's that 4 fused carbon ring thing called? Styrene skeleton, yes. What's it made of? Just carbon and hydrogen. Cholesterol, carbon, hydrogen, and 1 oxygen. That's it. Right? Where do you find cholesterol? Where is it? Where do you find it? Membranes, right? Some in adipose tissue, right? And what else is it used for? Making hormones, particularly the sex hormones, are derivatives of cholesterol. All right, now proteins. Macromolecular substances composed of amino acids. And if you ever get tired of saying amino acid, I guess you could always call them amino acids, but I don't like that pronunciation. Now, macromolecular, what does that mean? Many. Huge. I mean, we're talking about things that have typically molecular weights in excess of 10,000. Huge, huge molecules. All right, now, amino acids are the building blocks. They are made of amino acids. How? You string together one amino acid after another in a chain. And I will bet you couldn't guess in a million years how you string them together. What do you got to take out every time you add another amino acid on? Now, what's been the typical theme? Taking out what? Well, hydrogen's part of it. Water. It's a dehydration synthesis. And we're going to call these peptide bonds that we stick between amino acids. All right, now first of all, what is an amino acid? Well, it is a molecule that has 2 functional groups on it. 2, at least 2. There could be others, but these 2 have to be there. There's an amino group, that's an NH2, and there's a carboxyl group. We've seen that before. We saw that on the fatty acids. They have a carboxyl group on the end. All right? Now, what did we say about carboxyl groups? When you put them in aqueous solution, they can function as what? They're proton donors. What are proton donors called? Acids, right. Close. Acids. Now, the amino group. The amino group can act as a proton acceptor. Now, what was that? What did we call things that act as proton acceptors? Bases, right. So look at this. If an amino acid has an amino group on one end and a carboxyl group on the other, it can have the properties of both an acid, due to the carboxyl group, and a base, due to the amino group. And there is, as you might guess, a term. Amphoteric. So anything that is amphoteric has the properties of both an acid and a base. Okay? So amino acids are amphoteric. They have the properties of both an acid and a base. What part accounts for the acid properties? The carboxyl group. What part accounts for the basic properties? The amino group. Right. Okay, so there we go. Water also is amphoteric. Because water can, and it does, donate protons in certain circumstances, and in others it accepts them. And if a water molecule picks up a proton, what ion does it produce? Now remember, what's the formula for water? H2O. Throw a proton on. What's it going to be now? Hydronium. H3O with a +1 charge. That's it. So if a water molecule picks up a proton, it has given you an hydronium ion. Yes. All right. Very good. Here's the general structure of an amino acid. So here we have the amino group on one end. That can act as a base. It can accept a proton. Here's the carboxyl group on the other end. It can donate that hydrogen away. All right? Now, here in the center, there's a CH and the R. Well, what's the R? Well, the R is how one amino acid differs from another. In the simplest case, that R would be replaced with a hydrogen, an H. You know, in other cases, it'll have other functional groups attached. Okay? Here's an example. This is cysteine, and you notice in place of the R, there's carbon, 2 hydrogens there, and then a sulfur and a hydrogen attached. That's sulfhydryl group. All right, so this is a sulfur-containing amino acid. But let's just go back to this one. What elements do you find in an amino acid? What's up there? Carbon, hydrogen, oxygen, nitrogen. In every case, those are the elements that are going to be there. Carbon, hydrogen, oxygen, nitrogen. Now, some other things we can add. Like the one we had up there a while ago, the cysteine, you can also throw in a sulfur. Some of them contain sulfur. All right? Well, the keratin that you find in your hair, for example. Did you ever singe it? Did you ever singe your hair? What does it smell like? Kind of a sulfury smell, right? Guess what's in it? Well, that amino acid we just saw there a moment ago. All right, some others, like the proteins, casein that you find in milk, it also has phosphorus in it. All right, but at a minimum, carbon, hydrogen, oxygen, nitrogen, and then some will also have sulfur, and some will also have phosphorus. But that's it when it comes to the elements that make up an amino acid. All right, so continuing on, let's string them together. And when we start stringing amino acids together, we start forming what, first of all, we can call peptides. And a peptide is just simply formed when you put together 2 or more amino acids. And the simplest one, you put together 2 amino acids, you take out a molecule of water, you've got what we can call a dipeptide. It's 2 amino acids linked together by way of a peptide bond. So in a peptide or protein, between the adjacent amino acids, you're going to find peptide bonds. Peptide bonds. Now, how do we get them? Well, peptide bond formation is a kind of dehydration synthesis. You take out a molecule of water every time you stick two amino acids together. And if you add another amino acid on, you're going to have to take out another molecule of water, and so on down the line. And we're just stringing them together. It's beads stringing again, one right after the other, in what starts off as a linear chain. Peptides, when you hear that term at the cocktail party, it generally means fewer than 50 amino acids strung together in a chain. All right? If you've got 100 or more, you've got a protein. What's between 50 and 100? Well, it's no man's land. You can call it whatever you want. All right, so there you go. Probably called a polypeptide when you get between 50 and 100. But anyway, there we are. Peptides, proteins. Degree of size, how many you string together. You can name them, particularly the short ones, by how many amino acid residues. So as we said, like a dipeptide would be 2 amino acids strung together. Tri would mean 3. If you had a pentapeptide, that's 5 in a chain. I mean, I wouldn't worry about anything other than that. Not worth it. Here is a peptide bond in graphic form. So that is the residue of one amino acid. Here's the other. And right here in the center, that is the peptide bond. And what had to be taken out in order to form the peptide bond? Water. A molecule of water. Now, if you want to break them apart, what do you want to have to do with the water? Put it back. What is the term that we've used before that means breaking things apart by adding back water? Hydrolysis. Right. So if you want to ingest protein, and you do, and you want to absorb it and use it, you're going to have to hydrolyze it first into polypeptides and peptides, which are shorter chains of amino acids, and then you're actually going to have to cleave all those peptide bonds to get it down to the amino acids before you can absorb them. And we do that all the time. You know, they've got enzymes that facilitate that, as you'll find out when you talk about digestion in Biology 108. Now, structures of proteins. Here are some terms again. Stars need to go with these because we need to know what they are, because we use them now and forevermore. Now, we have a hierarchy of structures. Now, what do we mean by a hierarchy? Well, the parts all go together, right? In which some are built on others, right? The higher level ones are built on ones below them. Yes, but let's take a look at it. Primary structure. Now, you go to the cocktail party, there's a cluster of people over here in the corner of the room chattering about primary structure of protein. What are they talking about? Well, here's what it means. It means the number and sequence of amino acids in the protein. Now remember, we're chaining together amino acids, one right after the other in a linear chain. And every time you add an amino acid onto the chain, what did you have to take out? Water. All right, now, there are like 20 different amino acids. So there's a huge combination by which these things can get connected together. The length of the chain is important, so how many, and the sequence, in other words, the order in which they come. All right, that's the primary structure. That's all there is to it. How many, and in what order. All right, there you go. So if you take one of the amino acids out, and stick something else in its place, you alter the primary structure, right? Yes. Or if you chop 3 of them off and you don't replace them, well, you change the primary structure because now it's a different length from what was originally intended. All right. Everything else, as far as these structures are concerned, all the others are built on this. You change this, you change them. All right? What have we got here? Well, this is kind of a representation of it. And each of these little chiclet things would represent an amino acid, and the line between would be a peptide bond. All right, so there we go. All right, that's the primary structure. A linear chain of amino acids held together by peptide bonds. All right? Secondary structure. Secondary structure is the bending of that primary chain into one of 2 conformations. We can either have what's called a beta pleated sheet, zigzagging, take paper and fold it into pleats, right? That's kind of what it would look like. Or in an alpha helix. What's a helix? A spiral. All right, it's a spiral. So typically now, once you've got that linear chain, it starts bending and folding on itself and producing areas that are either kind of zigzaggy pleats or corkscrewy-like spirals. All right? There you go. What makes the difference? Depends on what kind of amino acids are there. If they're the short, small amino acids, they tend to give the pleats. If they're the bigger amino acids, they tend to form the helix, the spiral, or the coil sort of thing. All right. Why do we get it? Something that'll warm the cockles of your hearts. Hydrogen bonds. What's a hydrogen bond? How do you define that? Force of attraction. I'll start. Force of attraction between what 2 things? Why is it called a hydrogen bond? Hydrogen and unshared pair of electrons. Now, we've talked about this before where we have these forming between adjacent molecules. This forms within the same molecule because it's such a big molecule. The thing can fold and bend on itself, and you can get hydrogen bonds forming in lots of different locations because you're going to have hydrogens, and they're going to be unshared pairs of electrons on either oxygens or nitrogens, and they come together and they form hydrogen bonds. And that's what gives us this secondary structure. Okay? All right, so there it is. So the secondary is built on the primary. If you go back and put in a different amino acid here and there, you're going to change where those hydrogen bonds can form. So you might transform the thing from a pleated sheet into, if you put in enough, into an area that is, in essence, a helix. All right? Well, that can have implications on down the road, too. So, if we go to the next slide, here's kind of a representation of the pleated sheet, and the little red dots would be hydrogen bonds. And here is the spiral, and again, the red dots would indicate where hydrogen bonds are formed. And that's what's holding it either in the pleated sheet arrangement or the helix arrangement, the helical arrangement. Now, all of that leads to the next level, the tertiary structure. Now, once you've got those pleated sheet and helical areas, this whole thing now again starts folding and bending on itself. And again, hydrogen bonds are one of the things that can be responsible for that. And once again, where the hydrogen bonds can form depends on the amino acids that are there. So it's all built on the primary and the secondary structure. So if you go back and change the primary structure with different amino acids, you're changing the secondary and you're changing the tertiary. Why do we care about the tertiary? Well, for this reason. tertiary structure is the three-dimensional shape of a peptide. Three-dimensional shape. And when it comes to proteins and peptides, the shape determines the function. Another way you could write it, shape equals function. And if you alter the shape, you alter the function. All right? Now, another term to add to your growing lexicon, denaturation. Denaturation of protein. What does it mean? It just simply means altering the shape of the molecule. Altering the shape of the molecule. Not breaking it down, we're not hydrolyzing it, we're just simply altering the shape. And when you alter the shape, what happens to the function? It's changed. It may be lost or it doesn't work like was originally intended. Now let's take an example of protein denaturation. And I think I'll just sit down for this one. Get a chair. Okay, getting tired and weary. It's because I'm getting old, I guess. But anyhow. Now, you're all familiar with eggs, right? Plain old hen eggs, right? All right, now let's get a couple of eggs out of the carton. And one we're going to crack open raw and put it out in a saucer. And what do you notice? How would you describe its appearance? What do you see? Well, there's a yolk, right, near the center. It's kind of a yellow, orangey yellow color, right? Depending on whether they're free-range chickens or not, you know. But anyhow, and then what's it surrounded by? The egg white, right? Well, the egg white is albumin. It's a protein. And the molecules in that protein are like little balls, little spheres. And they are water soluble. Really, it's a solution of what's there. You know, it is viscous and slimy and all like that, but still, it's a solution. There's a lot of protein there, not a lot of water. Now, what was the purpose? What did Mother Nature have in mind when she put that stuff in the egg? What's it there for? To protect and nourish the embryo. That's right. I mean, you know, did you all ever see the movie The Coneheads? They were always consuming mass quantities of fried animal embryos. Remember that? Well, anyhow, I know I'm dating myself by mentioning them, but nonetheless I shall. All right, so there you go. Now, what if you took the other egg and plopped it in a pot of boiling water? It's going to come out hard-boiled, isn't it? Now, what's happened to the egg white? What's happened to it when you hard-boil the egg? Yes, it's now a solid, a white gelatinous solid, isn't it? You know what you've done to it? You've taken those little balls and you've unwound the things. You've changed the shape. You've denatured it. That's all you've done. Have you broken it down? No. It's still the same amino acids and, you know, original length and, you know, and sequence. We haven't done a thing there. All we did was change the shape of the molecule. Now, if you had those two eggs and you had fertile eggs, and the hen is sitting on the one that you didn't boil, and you slip back under her the one that you did, which one's going to hatch out the chick in a few weeks? The one you didn't boil, right? What about the one you boiled? Is that, are you going to get a chick hatch out of that? I don't think so. Because you've denatured the proteins that are in there. They're not going to work. All right? Now, that's all there is to protein denaturation. You have the same deal when you separate out your eggs and you're making a meringue or something and you get the mixer going. All you're doing is unwinding those little spheres of protein. You're just denaturing it. You're not changing, you know, the chemical composition of it in the least. You're just unwinding it. But I don't think you could turn that into a nice chocolate mousse and stick it back in the eggshell and get a chicken hatch out of that either. All right? Well, that's simple, isn't it? Yes. All right, so that's denaturation of proteins. All you're doing is changing the shape. All right, here we are. Here we have the protein folded up in its secondary conformation, and now we've got this overall shape that results when that folds on itself, and there it is. And they have kind of like a light blue around here, an outline that is depicting the shape. All right, now, all proteins are going to have a primary, secondary, and tertiary structure. But some have another level, and that's called quaternary. And that results when you take more than one peptide or protein and combine them together in some overall complex shape. Okay? A good example of something that will have quaternary structure. Hemoglobin. Hemoglobin has quaternary structure. It's made up of 4 polypeptides. 4 polypeptides combined together in a particular shape. And in order for it to function properly, it needs to have that particular shape. And if you alter that, well, it either doesn't work or it doesn't work very well. You know, here's an example. Most of the hemoglobin in your red blood cells is what we would call hemoglobin A, the adult form of hemoglobin. Okay? Some people may have another variant, and there are lots of variants of hemoglobin. There's one called hemoglobin S, as in Sam. Okay? That's the one that people with sickle cell trait or sickle cell anemia have, depending on the amount. Whether you got the trait or if you got more, you have the anemia. Now, on one of those peptides, in fact, it's actually 2 of them, because there are 2 peptides, and there's 2 copies of each, but anyhow, but nonetheless. There's one amino acid different from what you have on hemoglobin A, and that alters the primary, secondary, tertiary, and quaternary structure of that hemoglobin. So when you get hemoglobin S into areas like out in capillaries, the smallest of the blood vessels, where there's not a lot of oxygen, the shape of the molecule changes, it alters the shape of the red cells, they clog up the capillaries, they cause lots of problems. And they don't really carry oxygen as efficiently as hemoglobin A. So there you go. A difference in shape can have a profound difference in the way things function. All right? So, when we're talking about quaternary structure, we're talking about the overall shape that you get when you combine the 2 or more protein or peptide subunits together. And, again, shape equals function. So if you alter the shape, you alter the function. And if we take a look at this one, here we have, in this case, 2 peptides or proteins. There's one, there's the other, and they're combined together in this particular shape. And in order for this to have the intended function, that shape is very important. This might be an enzyme. And maybe the active side of the enzyme is the pocket between where those 2 proteins come together. It's usually just a small area. And if those don't have exactly the right shape, they're not going to have the pocket of the proper size and shape, so the thing might not work at all, or if it does work, it isn't going to be as originally intended. Most of the time it doesn't work as well. Sometimes it does work better. It doesn't happen too often, but sometimes. All right, so there we go. There's proteins. Simple. What are they? What are they made of? What are the building blocks? What are the building blocks of proteins? What were they called? Bless you. Amino acids. And what kind of bond do you find between the amino acids and a peptide or protein? What was it called? Peptide bond, right? And how did you form it? It's a kind of dehydration, right? So what are you taking out when you stick 2 amino acids together? molecule of water. And if you want to break it apart, what do you got to add back? Water. And what's that process called of breaking things apart by adding back water? Hydrolysis. See how easy that is. All right. Also, remember your primary, secondary, tertiary, and quaternary structures of proteins. Okay? And remember it's a hierarchy. So like quaternary is based on tertiary, tertiary on secondary, and all everything really on primary. All right, nucleic acids. These make up genetic information as well as functioning, as we'll see, in protein synthesis within cells. There are two major types, and you probably already know this. RNA and DNA. Now, RNA, those letters stand for ribonucleic acid, and DNA stands, of course, for deoxyribonucleic acid. Now, where do we get the names? Well, it's such an easy little thing, because let's move on to the next slide. All right. They're made up of building blocks. These are big molecules. Now, these can be just as large in some cases as proteins. So they're made up of building blocks. And all the macromolecules we've had were. The building blocks this time are called nucleotides. Nucleotides. So what are you going to do? You're going to string together nucleotides. I'll let you guess in a million years how you string them together. What are you going to take out? Of course, H2O. All right, here you go. Now, what makes up a nucleotide? Whoops. Okay. Nucleotides have three components. They have a sugar, they have a phosphate, and they have a base. Well, the sugar. It's either ribose or it's deoxyribose, depending on which kind of nucleic acid you're talking about. If you're talking about ribonucleic acid, all of the nucleotides contain ribose as their sugar. If you're talking about DNA, or deoxyribonucleic acid, all the nucleotides contain deoxyribose as the sugar. All right? And remember ribose and deoxyribose, how many carbons did you find in those? They're pentoses, right? Yes, they are. All right. So we have a sugar, we have a phosphate, PO4, phosphate group, then we have some kind of base, either a purine or pyrimidine. Now, there are only 5 bases. 3 of them are found in both. And then there's 2 others. You'll find one in DNA and the other one in RNA. They're easy. There's ribose. Oh, look at that hydroxyl group there in red. Isn't that pretty? Well, what's between this and deoxyribose? There it is. What have we got here now? We took out the oxygen. I wonder why it's called deoxyribose. We lost an oxygen. Yes, isn't that sweet? All right. Now, nucleotides. Sugar, phosphate, base. Doesn't matter whether it's an RNA or DNA. That's the basic arrangement. The only difference is, what's the sugar if it's RNA? Ribose. What is it if it's DNA? Deoxyribose, right? And then we'll see a little bit of difference over here with the bases. So let's get to that. Here we are stringing them together. This is a single chain. Here's a double. Don't worry about that. We'll talk about that when we talk about nucleic acids and protein synthesis. That we'll get to in metabolism after we've chatted about cells next Thursday. Now, here are the bases. These are the ones you find in DNA. These are the ones you find in DNA. Adenine, thymine, cytosine, guanine. Those are the 4 bases that can be found in the nucleotides of DNA. All right? adenine, thymine, cytosine, guanine. All right? So, if it's a DNA nucleotide, you'll have deoxyribose and phosphate, and then either an adenine, a thymine, a cytosine, or a guanine. There are just 4 nucleotides. That's it for DNA. So the only difference between the DNA in a rabbit and you is the sequence in which you put those things together. In fact, we could even make it more dramatic than that. I mean, you know, the difference between the DNA in you and a lowly amoeba depends again on the sequence of those things and how much there is too. But anyhow. Now, what about RNA? Well, if they're RNA nucleotides, what did we say was the sugar? Ribose. So we have ribose and phosphate. Now for the bases. Adenine, cytosine, guanine. And one more. There's no thymine in an RNA nucleotide. Some of them will have adenine, some cytosine, some guanine, and the others will have uracil. There's no thymine in RNA. Instead, it's uracil. It's U-R-A-C-I-L. Yeah, it's hard to see in here. You can't dim these lights, which is a bad thing. Uracil. U-R-A-C-I-L is uracil. Uracil instead of thymine. There's no thymine in an RNA nucleotide. All right? So in an RNA nucleotide, ribose, phosphate, and then either adenine, uracil, cytosine, or guanine. Okay? That's simple. Then if you want to make DNA, you take the appropriate nucleotides and you string them together. And same deal if you want to make RNA. You take the appropriate nucleotides that have ribose in them, and you string them together. Simple. All right? And these can be rather large, very large, in fact, particularly DNA, very large molecules. Now we've got one more to go, and that's this one. This is an energy compound. An energy compound. And this is the primo, I guess we could say, energy compound. ATP. And those letters stand for, as you can see down here, adenosine triphosphate. Adenosine triphosphate. Commonly just abbreviated ATP. Now, triphosphate. 1, 2, 3 phosphates. Neat, right? What do we need it for? Well, this is typically the molecule we have in cells for storing energy. This stores energy. When you make ATP, you store energy. When you need to use the energy, what are you going to do? You're going to break off one of those phosphate groups. The phosphate bonds are high energy bonds. When you form the phosphate bonds, you store energy. When you break them off, you release it. And typically, we're only breaking off one of them, as we'll see when we get to metabolism. All right, so what have we got? Well, we've got ribose, we've got adenine, and we've got these 3 phosphates. That's all there is to it. How many phosphorus would there be in a molecule of ATP? 3. Yes, and what other elements are there? Carbon, hydrogen, oxygen, nitrogen, and then 3 phosphorus. And that's it. Now, isn't that simple? Yes. Yes, yes, it is. All right, so ATP, that's coming back to visit. You'll enjoy it immensely. All right, so Carbohydrates. Carbon, hydrogen, oxygen. What else can we say about that? What's the hydrogen-oxygen ratio in a carbohydrate? 2 to 1. All right. Simple sugars, disaccharides, polysaccharides, right? Remember those? All right. Lipids. Carbon, hydrogen, oxygen. If we're talking about triglycerides, that's all that's there. What about the hydrogen-oxygen ratio, though? Certainly not 2 to 1. A lot more hydrogen than you're going to find anywhere else in carbohydrates. And things like triglycerides, glycerol, fatty acids, and so on. And then phospholipids, add to that phosphorus at least. Carbon, hydrogen, oxygen, phosphorus, maybe nitrogen, right? Okay, how many phosphorus in a phospholipid? Just one. Okay. All right. And then there's cholesterol, carbon, hydrogen, 1 oxygen, right? Okay. What do you use cholesterol for? Sex hormones, cell membranes, right? Phospholipids, cell membranes primarily. What about triglycerides? Where'd you find those? Adipose tissue, stored in adipose tissue, yes. Proteins, carbon, hydrogen, oxygen, nitrogen, sometimes sulfur, but also we could say sometimes phosphorus. How many? Gobs of carbons, hydrogens, oxygens, and nitrogens, remember. Because what's a protein made of? Amino acids. Lots of amino acids strung together. All right, where do you find them? They're in cells. Enzymes are all proteins. Most of cell structure is protein. Okay? Nucleic acids. Elements, carbon, hydrogen, oxygen, nitrogen, and phosphorus. And there you go. And now what about energy compounds? What was the one we had there? ATP. Yes, how many phosphorus in ATP? Yes. All right, so without further ado, let's go to our little review sheets. And let's take the 2-pager first. Looks like multiple choice things. Got that one handy. Well, let's take a look at it. I think I'll be lazy and sit down. I don't rush show up on the thing I'm sitting on, but anyhow, they can be here. All right, well, let's take a look at number 1. Okay, which of the following is the storage form of excess glucose in the body? Here are the choices. A, amylose. B, cellulose. C, glycogen. D, cholesterol. E, peptides. Let me read them again. Storage form of glucose in the human body. Amylose is A. B, cellulose. C, glycogen. D, cholesterol. E, peptides. I see a large molecule is made primarily in liver and muscle cells. Glycogen, yes. All right? Oh, that's simple. Why is it not amylose? Glycogen's an amylopectin. You know what that means? Amylopectins are branched chains of glucose. Amyloses are linear chains, only plants make those. What about cellulose? Where do you find that? Plants, right. All right, let's go on to number 2. Which of the following terms refers to molecules that contain unequally shared electrons? A, salts. B, polar molecules. C, nonpolar molecules. Polar molecules, right. Partial separation of charge, the dipoles. You got that because something has a higher electronegativity than something else. All right. 3. Which of the following covalently bonded molecules are polar? Well, let me see if I can roll quickly up here on the board. Okay, let's see here. All right. All right. A, B is, C is, B, and E. All right. What are my bids? Polar. We want which ones are polar. A and E is correct. All right. The others are not. B, remember, is tetrahedral, and C and D are linear, but they're two atoms of the same element, so therefore the same electronegativities. All right. So there you go. 4. Which of the following is an inorganic compound? A, sucrose. B, cholesterol. C, amino acid. D, sodium bicarbonate. E, fatty acid. Which one is inorganic? Sodium bicarbonate. That's an ionic compound, yes. All right, number 5. Which of the following is true of an acid? A, they release hydroxide ions when in aqueous solution. B, they are proton acceptors. C, they cause the pH of a solution to increase. D, they generate hydronium ions when in aqueous solution. E, the hydronium ion concentration equals the hydroxide ion concentration. D as in dog, yes, they generate hydronium ions. They give off protons, water picks it up and generates hydroniums. E, if they have equal hydronium and hydroxides, that's neutral. That'd be a neutral solution. All right. Now what happens to pH when you add acid to something? Decreases, yes. All right, number 6. The substance is found to be composed of molecules containing carbon, hydrogen, and oxygen. One molecule contains 6 carbon atoms, and the hydrogen and oxygen atoms are in a 2-to-1 ratio. The substance is most likely which of the following? A pentose. B, an amino acid. C, a fatty acid. D, a monosaccharide. E, a nucleic acid. Monosaccharide would be the best. Why is it not a pentose? How many carbons did it have? 6. Pentoses only have 5. All right, we know it's not an amino acid because, you know, the right mix, right? We only had carbon, hydrogen, oxygen here. What else is an amino acid besides carbon, hydrogen, and oxygen? Nitrogen, right? Yes. Okay, see? There you go. All right. Think of what's in these things. You know, which elements are in them and then in what kind of ratios. All right, number 7. Which of the following best describes the composition of a triglyceride? Oh, this is an easy one. A, glycerol and 3 fatty acids. B, sugar phosphate backbone with 2 amino groups attached. C, 2 monosaccharides. D, 5 amino acid residues in a chain. E, glycerol, 2 fatty acids, a phosphate, and an amino alcohol. A is the best, yes. glycerol and 3 fatty acids. That's what it is. All right, now, 8. A substance contains an amino group and a carboxyl group. It does not contain any peptide bonds. The substance is likely which of the following? A, monosaccharide. B, an amino acid. C, a peptide. D, a phospholipid. E, a fatty acid. B is in boy, an amino acid. There's no peptide bond, so it could be a peptide. And it could be any of that other stuff based on really what we have. All right, 9. The class of lipid that is used as the basis of the sex hormones is, well, I don't think I have to read choices. What is it? Cholesterol, right. And 10, dehydration and hydrolysis are essentially opposites, which is used in the synthesis of a protein. Dehydration, absolutely. All right, now let's take a look at these. And we got a few minutes left. Get through this quickly. All right, now I'm going to give you these molecular formulas, and then what you need to do is just tell me, is it likely to be a carbohydrate, a lipid, a protein, a nucleic acid, or an energy compound? I mean, pretty simple. All right, so number 1 is C620H890O52 and N100. And 2 is C6400 H5000 O900 and 580 P300. 3 C310 H620 O310. 4 C58 H8609 and 5 C8 H22O18 and 4. Okay. Well now, what do you think number 1 might be? Yeah, it could be a protein or a peptide, I would say. It's probably the best answer for it. Because all we've got is carbon, hydrogen, oxygen, nitrogen. It's hardly a 2-to-1 on the hydrogen, oxygen, and there's quite a bit of nitrogen there, so there you go. I would say that's the best one. What about 2? There's probably a couple. Nucleic acid is probably the best. Possibly a protein, because it's awfully big. Okay, that's a lot of phosphorus in it, though. But it's possible that nucleic acid is probably the best answer for that one. All right, what about 3? That's a carbohydrate. Look at the hydrogen-oxygen ratio there, 2-to-1. What about 4? It's some kind of a lipid, probably a triglyceride. Okay. What about 5? The P3 is the giveaway. It's an energy compound, that's ATP. Okay? All right, as far as the chart's concerned, I mean, you can kind of fill that in. I mean, like, carbohydrates, lipids, proteins, nucleic acids, energy compounds. What's their function? What are some of the characteristics? Thinking in terms of what elements you got, ratios, where you can find them in the body, and then one example. I mean, there's lots of things you can fill in for some of that stuff. So, there you go. So, next Tuesday then, assuming that the snow and everything holds off, I think it's supposed to turn to rain. Test, majority of questions, multiple choice, but maybe something kind of like what I have here. You want to think about it. Most. Some. And then 5 or so short answer things, some which have multiple parts. They're not hard. You've got the whole time. When you get done, that's it. That's all we'll have that day is the test. Okay? And as far as lab is concerned, for me, I think both labs will really be on their second week on tissue. So the lab practical will not be next week. It'll be put off after because we haven't got everything done. Okay? So we're just kind of backing everything back, and that's what we'll do if, the weather interrupts. But I already have the test ready. It's in the print shop, so it should be ready for you. And periodic table, yes, I will supply it. It's like the one I gave out and we use in class, and it's in color. It should be. I'll find out about that. I can run them off in color if I have to, but anyway, so there you go. Enjoy. Keep warm. Thing tipped down on me.